A cell membrane, like you guys said as we were going through the homework, it is a lipid bilayer, specifically a phospholipid. Phospho meaning that it's just a lipid with phosphorus attached to it. The lipid bilayer is important because it forms the boundary of the cell. It's like the skin of the cell.
So the outside of the body.
That's right. And it kind of makes sense because if you think about lipids, what is another word we said for lipids? Fats, oils, right? You think about
Hydrophobic.
fat and oil. How does fat and oil interact with water?
Hydrophobic.
It does not.
Hydrophobic, it floats.
Yeah.
It doesn't mix, right? They repel each other. Exactly. So with a lipid bilayer, you basically have this kind of like layer of fat or this layer of oil. And so it repels water, keeps water from traveling through it nice and smooth. That way it can kind of like separate the outside from the inside. So if we do want to move things in and out of the cell, we have these channels. These are usually made out of proteins. So cell membrane, fats, proteins, sometimes cholesterol as well. Right, those are the sort of things that you'll find there in that so-called skin of the cell.
So,
Yes?
cell membrane is skin? No.
It is the outer covering of the cell. So that's why I call it like the skin, right? The skin is the outer covering of your body. If you look at a cell, the cell membrane is the outer covering of the cell.
So cell membrane is synonymous with skin? So
No,
No.
the membrane is not skin. But it
The
behaves.
outer covering might be a part.
It's an analogy, right? It's not going to be exact. It's not one-to-one. But the same way that the skin covers our body, the cell membrane covers the cell.
Oh, that's
It- It's not like the cell membrane is going to have hairs and sweat glands and things like that coming out of it, right? It's not the skin,
no, okay.
but the same way the skin covers
I
us.
understand now. So I thought you was comparing it to our skin.
So, just analogy, similar function. Okay. Inside the cell, right? So in the deepest part of the cell, we have the nucleus. This is separated from the rest of the cell by the nuclear envelope, which is once again very similar to the cell membrane. Okay, so also made of lipids. Inside is what we call nucleoplasm rather than cytoplasm. Remember, cytoplasm is stuff outside the nucleus. So inside the nucleus, it is nucleoplasm instead. And the nucleoplasm is where we have things like the DNA. Now, DNA takes different forms. It has different shapes depending on the life cycle of the cell. For example, while the DNA is in the nucleus and the cell is behaving normally, we want to be able to access the DNA, to be able to look at the code of the DNA, copy that information, make proteins, make molecules, do cell functions using the information in the DNA.
Not us, but the body,
Well,
right?
okay, the body, the cell itself. So, in order to access this DNA information, well, we need to be able to easily access the DNA, right? It's like if you're constantly going to that cookbook, right? We use the cookbook analogy for DNA. So if you're constantly referring to the cookbook, you don't want this cookbook stored on the top shelf of a 12-shelf bookshelf, sandwiched between all these other thick books, because it's going to be really hard to get to every time you want to look up a recipe. You want it somewhere that's easy to access, maybe even already open to the page you want, right? So the body, the cell, also wants its DNA to be easy to access while it's in its normal state. So this form of DNA that's easy to access is called chromatin. Chromatin is a relaxed form of our chromosomes. So the chromosome is DNA that's tightly bound together. It's all smushed and compact. But if it's smushed and compact, it's hard to access this information. Chromatin then is this chromosome but relaxed. So it's no longer all tied and strung up together. It's relaxed, it's free, and it's easy to access what you need to. Yes, Jay?
Is chromatin considered to be the protein then?
So
Because in the textbook it said that nucleoplasm was a protein-rich environment.
Mhm.
So,
chromatin itself is not a protein. But chromatin is DNA still.
okay.
Now you do have other proteins in the nucleoplasm, but chromatin itself would not be an example.
So we don't have to know anything like that? Okay.
You don't need to worry about specific proteins.
Too deep, too deep.
So you're saying that instead of the DNA being like the way it is supposed to be, it's more of a fluid?
Right. So when we talk about DNA, we're like, okay, it's this double helix, right? And we always draw this as this nice, long, straight strand. But in reality, we take this strand of DNA and we kind of like tie it up like a ball of yarn or something. Right? So it's all tight and compact because, I mean, space is kind of precious. Right? So we kind of squish it all together. But that's only in situations when the cell is ready to divide. When it's ready to divide this information into two pieces. Right? Under normal circumstances, when the cell needs to look at this information, it doesn't want everything tied up together. It wants it in that nice line that we see in textbooks. Right? So when it's nice and open and easy to access, that is our chromatin. When it's all wound up together, that is our tight chromosome.
Chromatin were like inside the nucleus or?
Yes, so chromosomes are kind of like our groupings of DNA, so yes, those will be inside the nucleus.
Which one is tight and compact?
So chromatin is going to be the loose one.
Okay.
So when we talk about our chromosomes or our chromatids, that's going to be when we have the tight, compact DNA.
So the chromatin?
So chromatin is loose, and then chromosomes or chromatids with a D is going to be compact.
Oh, okay, I just wrote it down. Compact and tight. Gotcha.
Okay. Inside the nucleus, we have the nucleolus. This nucleolus is important because this is what creates that rRNA for the ribosomes. Nucleolus creates RNA used to create our ribosomes. Also creates and stores the proteins used in our ribosomes as well. Moving on, we have the cytoplasm. Okay, so everything we just talked about, right? Nucleus, nucleoplasm, nucleolus was all inside this nucleus here. Now we're going to talk about the stuff outside the nucleus, right? So in the cytoplasm surrounding the nucleus, we've got water, proteins, carbs, lipids, salts, just all sorts of things floating in that fluid. Okay. Most of the cell functions happen in the cytoplasm, right? That's where most of the action is. Nucleus is primarily just storing that DNA. So, what sort of things can we find inside the cytoplasm? As far as organelles go, like cellular structures, we have a whole bunch of things. General structures are things like tubules, which are small cellular tubes. Vesicles, so these are small sacs, small pouches that contain some sort of liquid. Granules, these are small particles without membranes, right? So without that cell membrane, right, that lipid membrane. Fibrils, these are fibers or strands. So these are general structures that you'll find. And then as far as specific organelles, like actual, like complex cellular components, we have things like the ER, the Golgi apparatus, mitochondria, lysosomes, ribosomes, and centrosomes. So let's talk about some of these organelles. First one is the endoplasmic reticulum, the ER. This is basically the highway system or the transport system of the cell. It's a network of tubes and vesicles. So it's basically just a bunch of pipes and a bunch of bubbles that it uses to transport, the cell uses to transport things around. Like Super Mario, exactly. Okay. One of the interesting things about the endoplasmic reticulum is that it can be rough versus smooth. Rough means that it's got some extra bumps on it. These bumps are ribosomes. And this makes sense because we mentioned ribosomes are the location where what type of synthesis occurs. What do we make in the ribosomes?
Protein.
Proteins, exactly. We make proteins in the ribosomes. And then we need to move the proteins to where they're going to do their work. So it makes sense that our ribosomes, our factories, are located right next to the highway, right, to this transportation network. So as soon as they make the proteins can go through here and travel to the correct location. So, ER, you've got rough, which includes ribosomes, smooth, no ribosomes, and it assists with the transfer of just molecules throughout the cell.
So what's the smooth?
So smooth is just transport. There are no ribosomes attached to it.
Okay.
But you said smooth for transfer of molecules, sir. What molecules?
Any molecules. It could be
Nourishment.
RNA, it could be proteins, right? Just things that the cells need. The ER helps to move it around.
So the smooth also does that, and the rough also does that.
Yes, they both do that. The only
But rough
difference.
has the ribosome, but smooth doesn't.
Correct. Okay, our next organelle is, rough versus smooth. So rough, okay, surface is rough. Why? Because there's ribosomes. Okay, smooth, surface is smooth, no ribosomes. Okay, so rough, right? Usually because we need to move a whole bunch of proteins around. Smooth, don't need to move as many proteins around. Remember that aside from being on the endoplasmic reticulum, the ribosomes can also be floating out in the cytoplasm by themselves. Okay.
You said the rough moves more proteins?
Mm-hmm. Yeah. Especially here, right, it mentions that if the cell is concerned with producing proteins that are used outside the cell, like in the rest of the body, right, it needs to get the proteins out of the cell. Okay. You'll also see a lot more rough ER. Speaking of moving things out of the cell, here we have the Golgi apparatus. So this is kind of like the post office of the cell. Basically, you have things coming from the endoplasmic reticulum, right? They're being transported around, and some of them get sent here to the Golgi apparatus. The Golgi apparatus is like FedEx or like UPS. Right? They take your stuff, they put it into a box, and then they ship it out. Okay? Maybe like international mail, right? They ship it out. So they package items into the boxes. In this case, these are the vesicles, the small sacs inside the cell. Okay? And then, as you can see, right, it can send these vesicles out of the cell to send these items out of the cell. It can also slightly adjust the things that you're sending as well. So if you're trying to send a protein, it can like stick a carb onto your protein to make a glycoprotein. Not that important. This part's not that important. General idea though is that it is used to transport things, like to package them up and transport them outside the cell. Okay. Next up, the mitochondria. Okay, this is the powerhouse of the cell. That's pretty much all we need to know. Actually, just one more thing, I guess.
ATP.
ATP. ATP, this is the energy currency of the cell.
How in depth are you gonna go gonna be asking specific things about, or just like what it does?
As far as the organelles, probably more general, more definition type.
It's not gonna ask about oxidation or like.
No, I'm not gonna ask you about like ATP in the Krebs cycle or anything like that.
Isn't that where the? Sorry.
Like aerobic versus anaerobic respiration. Don't worry.
Isn't
about
this the most radiosensitive part of the cell?
it. The most radiosensitive part?
Oh no.
I would not say that, no.
No.
Isn't that like
Part of it
stem?
depends on how you define radiosensitivity.
Then
But I would say the most,
what about the free radicals then?
so I would say the most radiosensitive part is gonna be your target molecule, which is DNA.
Okay.
Yeah.
DNA is gonna be your most radiosensitive. Because if you talk about radiosensitivity, usually it's gonna be some combination of how easy is it to affect and how much of an impact will it have on the cell. For example, let's say that radiation affects a vesicle and it destroys the vesicle. Well, that sounds bad, except for the fact that you've got thousands of vesicles in the cell. Losing one vesicle is not a big deal. But if the radiation hits the DNA and it destroys part of the DNA, well, now that is a big deal because you don't have another copy of that, and now the cell may lose its function.
But so there's DNA in the organelles and then there's DNA in the nucleus. Like there's
So
DNA
the
here.
only other organelle that has DNA is the mitochondria. There's kind of this fun fact where the mitochondria's DNA actually comes from your mother's side. It's just been passed down through your mother's generation since humans were created or born or evolved or what have you. So mitochondria have their own kind of like unique DNA. Yes, that's true. But once again, you've got a bunch of mitochondria in the body. It's not a big deal if one or two fail to function.
Okay.
It is a big deal if your only set of DNA in the nucleus fails to function.
Okay.
So as far as radiosensitivity go, I would probably say your nucleus DNA is the most sensitive.
Okay.
So
Yes.
the statement that DNA is only in the nucleus is not correct, sir?
So generally speaking, DNA is only in the nucleus. When we talk about human chromosomes. So the 23 human chromosomes, right? 23 pairs of human chromosomes, those are only in the nucleus.
So this DNA is where, sir? In the mitochondria.
So mitochondria has its kind of own unique DNA. Okay. The idea behind this is that we suspect way back when humans evolved or when cells first evolved, the mitochondria was actually its own bacterium that kind of got sucked up by another cell, and it just kept its own DNA.
So what is this?
This is the mitochondria. This
Yeah.
is the powerhouse of the cell.
Mhm.
It uses energy in the form of ATP.
All right.
That's it.
That's what you need to know.
All right. Thank you. That's it.
Mhm.
What about stem cells?
Stem cells. Yes, I think we're going to talk about stem cells later.
I thought those were the most radiosensitive cells.
We are going to talk about those later. Yes.
See, Johns. You know, we're
Mhm.
getting there. We're
Please
getting
look
there.
forward to it. Okay, nice. So ATP, this is our energy currency, our energy molecule that mitochondria deals with. Okay, you got this thing, you pop off a molecule, it gives you energy, you stick the molecule back on, it stores energy. Nice and easy. Lysosomes. Lysosomes, their name kind of gives away what they do. What does it mean for lysis? Lyse?
Destroy.
Nasty.
Destroy, yes, not
Lysol.
nasty, but
Mhm.
destroy. Right, like Lysol, right?
Yeah,
Destroys
Lysol,
germs.
yeah.
Destroys 99.9% of all germs.
Mm-hmm.
So, lysosomes destroy things. So this is our garbage disposal. Right? It destroys unwanted proteins, unwanted molecules in the cells. So it's basically this sac that's once again surrounded by a lipid membrane. Inside are chemicals that are very reactive and that will destroy things. So you take what you don't want, throw it inside, the chemicals break it down, and then you have your raw ingredients to create more stuff with. Problem, stuff inside here is toxic. So if the membrane breaks for whatever reason and this toxic stuff enters the cell, it can start to break down the cell itself. Okay? So that's why the cell tries to keep it nice and enclosed in that lipid layer. Now, radiation can break through the lipid membrane, so radiation can cause the lysosomes to rupture and for the contents to start attacking the rest of the cell. Yes?
And they see that in anaphase, right?
So this is just general. This does
Okay.
not.
So that's just
correspond to
apoptosis.
anything. Apoptosis may also involve the cell using lysosomes to destroy itself. Okay, ribosomes. We've talked about these before. These are just our protein factories. They take a chain of mRNA, which is the instructions. They find the corresponding tRNA. tRNA is attached to amino acid, drag it over, chain them up together, and that is your protein. Okay. Centrosomes. Okay, these are kind of important because we are going to talk about them in context of cell division. These act like a winch, right? They act kind of like a crank. And they are used to pull our chromosomes apart during cell division, okay? Or specifically our sister chromatids apart during cell division. Okay, so we have chromosomes, we create copies of them, we line them up in the center of the cell, we attach them to a bunch of strings called microtubules, and then we use them to pull them apart. That way, each half of the cell gets its own chromosome. Then you chop the cell in half, and now you have two cells. Okay, so centrosomes used during cell replication, cell division. Okay, and so that brings us to cell division. There are two types of cell division. As we see here on this diagram, they are mitosis and meiosis. Let's see. So Janelle, how are mitosis and meiosis different?
So they're separating into. Hold on. Is it like the daughter cell? It says, okay, sorry.
Well, that's okay. So keep going. So you said something about daughter cells.
Split one, splits into two.
Mhm.
And meiosis has there's two phases of it.
Very good.
And
So after two phases, how many cells do we have then?
so
4 of them, exactly. So mitosis, you end up with 2 cells. Meiosis, you end up with 4 cells. Why the difference? It's because, Charo,
One is germ cells and one is
germ cells, you mean that our body's getting sick?
no,
Oh, okay, what kind of germ cells are we talking about?
like female or male productive cells.
Yes, our reproductive cells. Germ cells are reproductive cells, sex cells, right? So somatic cells, body cells, mitosis, germ cells, reproductive cells, sperm, egg, that is meiosis. Okay, so body cells divides only once, so 1 cell becomes 2. In meiosis, sperm and egg, they divide twice, and so we end up with 4 cells. Okay, the scientific names for our germ cells here, rather than egg and sperm, it is oogonium and spermatogonium.
Oh wow, okay. I got that question wrong.
me too.
What in the
Yes.
elsewhere it
Yes.
says ovum and
Mhm.
spermatozoa.
Uh-huh.
So will you take those as well?
Yeah, that's what I put.
So
I
let's
put
talk about
ova
that.
and ovum
That's
and
what I did
sperm
too.
and
I
spermatozoa.
remember writing the
All
over.
right, let's talk about it. Let's talk about it in just a moment. Just a moment.
Oh,
Hang
question.
on to that for a little bit longer. Yes.
Somatic, you said that was the body cells? No?
Somatic is body cells.
Okay,
Yes.
gotcha.
So you see the word somatic?
Mm-hmm.
It refers to a body cell. If you see the word germ, it refers to a sex cell, either sperm or egg. So as an example, liver cells are
Somatic.
somatic. Skin cells are
Somatic.
somatic. Okay, sperm cells are germ cells. Right, brain cells are somatic, right? Just like that. Okay.
Just like that. See?
So, mitosis. So this deals with somatic cells, body cells, any of your body cells, liver cells, intestinal cells, right? In this case, we have a parent cell, that's our original cell. It is going to divide one time, so it divides, and now we have two daughter cells. These two daughter cells are going to be identical to the parent cell, right? It's like clones. Okay. The cellular material is going to be split evenly, or as close to evenly as possible, between the two cells. So there's going to be ribosomes. The cell's going to try and divide the ribosomes equally, right? There's going to be like your lysosomes, divide that equally, right? Now, ER, try and divide the ER up equally. Right? It's not like when you like cut a cake and one person gets 80% and the other person only gets 20%, right? That's not equal. But body does try to divide this equally. How does it do this? Well, cell division has 4 phases. Okay? It has the gap 1 phase, the S phase, the gap 2 phase, and the mitosis phase. These first 3 phases, gap 1, DNA synthesis, and gap 2, these 3 are referred to as interphase. Okay, so these 3 together are part of interphase. This part is important because we will be discussing these phases in later chapters. Okay, so this is something that you will want to know for later chapters. So gap 1, DNA synthesis, gap 2, and then mitosis. The first 3 are interphase, and then the mitosis phase can be further subdivided into more phases as well. Now, looking at those first phases, G1, gap phase. Why is it called a gap phase? Is it because it sells clothing and like baby apparel?
Ooh.
Right? I think that's what the gap sells. It's been a while since I've been to one. Okay, no, the gap 1 phase is basically a resting phase. So we also call this the pre-DNA synthesis phase because this happens before we create more DNA. Basically, the cell's kind of resting, it's just doing its normal job, not doing anything too fancy. It is going to be working on making more organelles in preparation for division though. Okay, so not too much happening during G1. S phase, this is when a lot of the action begins to take place. In the S phase, the cell is like, okay, enough procrastination, it's time to get ready for cell division. So I have my DNA, but I only have 1 copy of this DNA. If I want to divide into 2 cells, I need a second copy of this exact same DNA. And so the cell is going to copy, line by line, every single base pair on that DNA and create a completely new chromosome that looks exactly the same. And so we call these, right, our sister chromatids. Okay, so inside the cell, we have homologous chromosomes, basically one from the dad, one from the mom. Right, so this is chromosome number 1. I have a pair of chromosome number 1, right? One being from dad, one being from mom. I'm going to take this pair of chromosomes and I am going to copy them. So this information from the dad side, I need a second set of this information. This information from the mom side, I need a second set of information. So once we copy these chromosomes, now you see that I've got dad side plus a second version of the dad side. Right? Exact same information. I've got the mom side and a second copy of the mom side. Exact same information. So these are called sister chromatids. Okay? So these 2 perfectly equal sets of information are sister chromatids. So they form this kind of X shape, this central part where the X crosses, we call that a centromere. Now, this process also very important because depending on when DNA damage happens, it will affect what happens during this process as well. Okay, so make sure you do know this for when we go into the later chapters and talk about DNA damage. Yes.
Is centromere an actual organelle? Is
So,
that-
in this case, no, centromere is not an organelle.
it's just an addendum to a process?
Right, so this is just the central part of our chromosome.
Okay.
Okay, this is as opposed to a centrosome, which was that organelle that
Mhm.
acted as the bridge. Okay, so after S phase, we now have 2 copies of every single chromosome in the cell. Right, on the dad side, we have 2 copies of that one. On the mom side, we have 2 copies of that one. And we did that for all 23 pairs of chromosomes. So now we are prepared. We have all the information we need for cell division. The only thing left to do is to wait. So when we wait, we are entering G2. This is our second gap phase. So during this gap phase, now the cell is doing its final checks. It's like, okay, let me just double check the DNA, make sure I copied everything correctly. Let me make sure that I have enough stuff so that when I divide, I can still survive, right? Make sure I have enough ribosomes, make sure I have enough proteins, make sure I have enough organelles to survive when I duplicate. Okay, so G2 phase is kind of the preparation phase, right? The double checking phase. And then after G2, we will enter actual mitosis. Okay, so that what we just discussed, G1, S, and G2, all 3 of those make up interphase. This is the primary state of the cell. The cell is usually going to be in interphase. So cell spends most of its time here in interphase. All right. So when the cell is ready to divide, when we're ready for mitosis, we have 4 more phases to look at. They are prophase, metaphase, anaphase, and telophase. So these 4 phases make up the mitosis phase of cell division. Prophase, the very first phase. In this situation, in this phase, the nucleus enlarges and then begins to disappear. Right? So now we lose that nuclear membrane. The nucleus is no longer separated from the rest of the cell. But this makes sense because we now need to get the DNA out of the nucleus so that we can move it into separate cells. So we're going to destroy or dissolve the nucleus so the DNA is free. At the same time, the DNA, it used to be in chromatin form, right? Loose, able to be accessed. Now the DNA scrunches up, it curls up, it coils up into those chromosomes, like those tight bundles of DNA. You can't access it anymore, you can't read the information. Okay? So you can't really do anything with the DNA right now as we begin cell division. And finally, those centrosomes, right? Those winches, right? Those move to opposite ends of the cell. They move to the poles of the cell, right? It's like North Pole, South Pole. Okay, so they move to opposite ends of the cell so that they can pull the chromosomes to their side later on. Okay, so during prophase, this is what happens. Nucleus dissolves, DNA scrunches up, and then the chromosomes move to the ends. Metaphase. So meta, to me, meta sounds like middle. So this is the phase when everything lines up in the middle. Basically, all of our chromosomes that we got all go here to the center of the cell. It's like the equator of the cell. Our centrosomes here, right, send out these spindles, send out these wires that attach to these individual chromosomes, right? And then they're going to do tug of war where they pull on the chromosomes, and the chromosomes will travel towards those centrosomes. These wires attach to the center of the chromosome. Earlier, we said that the center of that chromosome, right, the place where the X crosses, is called the centromere. Right? So those spindles attach to the centromere, that center crossing point of our chromosomes, of the sister chromatids. At this point in metaphase, because the chromosomes are so tightly bound together, they are actually visible under a microscope. You can see with your eye the 23 chromosomes of the body. And in fact, if there has been any damage, any significant damage to the chromosome, like part of the chromosome has broken off, you can see that as well under a microscope. So chromosomal damage here is actually visible to the eye. Once we finish lining everything up, we move into anaphase. Anaphase is where we begin to split the cell in half. So, in metaphase, we lined up our sister chromatids. Remember, right? For moms, right? One copy of moms, second copy of moms, right? For the dad, one copy of dads, second copy of dads. Those are our sister chromatids. And so we pull them apart, right? So now, half of these go one way, half goes the other way. So this half is now getting a copy of moms and a copy of dads. This half is getting a copy of mom and a copy of dads. Okay? That way, each half will have the correct number of chromosomes. Okay. So the sister chromatids split apart. Half of the sister chromatids go one way, the other half go the other way. They end up in different cells.
Mr. Phung,
Yes.
we had said that cytokinesis appears in telophase, but it's already starting in anaphase. The
So you're talking about the splitting of the cell here?
Yeah, like this sheet.
Don't worry about that.
So
Just associated with telophase.
But did you not say that it happens in telophase?
Associated with telophase.
Oh, I see.
Okay, it's primarily a telophase thing. Okay, speaking of which, telophase. Okay, this is our cytokinesis. A cell pinching into two. So it's at this point where the cell actually begins to break apart into its two halves. Okay. At this point, at telophase, we've gotten everything into the correct sides of the cell. Half of everything went to the left, half of everything went to the right. So now we are able to chop the cell in half. Right? Cytokinesis is how we chop the cell in half. At the same time, because we have all the DNA on the correct side, our DNA can now begin to go back inside the nucleus. So the nucleus begins to reform. Okay, once the nucleus reforms, DNA relaxes, becomes chromatin again, and you have two normal cells. Okay. Remember, in this original cell, right, in this original cell, how many chromosomes did we start with?
23.
23 what?
23
23 pairs, right? So 46 total, right? 23 from the mom, 23 from the dad. After the S phase, right? Synthesis. How much DNA, how many chromosomes do we have?
Should be 46 pairs.
No, it's still 23 because they're attached at the centromere.
It is. So it's basically doubled,
Yeah, it should be
right?
46.
So it's basically doubled, right? Because now you have two copies,
Each.
right? So you used to have 23
Oh, yeah.
pairs. Now you have two copies for each of those pairs, right? As our sister chromatids.
So we
Okay.
get 46 each?
Say that again.
23 pairs.
So,
Should be
so we have basically 92 sister chromatids, right? Because we have 46 pairs,
Multiplied by 2.
right?
46 pairs. It says
It's
46
40,
Yeah.
46 pairs is 96.
Okay, yes, so 46 pairs, right, which ends up
Or
being 96.
92. 92.
Or,
So
sorry,
what-
92, yes,
92.
92
What
individual.
number we at?
Yeah.
Is it gonna be 46 or 92?
It's okay, right?
I can't do
Listen,
this.
work
Alright,
with
so
me.
come back here,
I
right?
think we need a
Alright,
break.
so let's stick with 46 pairs, right?
Okay, thank
Come
you.
back here, 46 pairs, right?
This is the last
Okay.
one.
Here, we now divide those 46 pairs in half, right? So 23 pairs go one way, 23 pairs go the other way. So in the end, how much does this cell have? 23 pairs.
23 and 23.
23, right? So 2 new cells that
Exactly
exactly
same.
mimic our original one. All right, you guys are right. Let's take a break. So time is 12:56.